Hello, my name is Billy Dunn. Today is June 6, 2018. This is the lab exercise video for biochemical testing for microbial identification by carbohydrate fermentation testing. The purpose of this test is it's another technique to identify unknown microorganisms by the fermentation technique, which occurs when the carbohydrates are broken down anaerobically. So I took 6 tubes 24 hours ago. It had a solution that was red, like the handle on my scissors. And then I took glucose powder and added half to each, which I labeled Staphylococcus epidermidis and Staph cerevisiae. And so I added that, shook it around, let it dissolve, and then the next step was to add the mini or micro Durham tubes inside, which you can see here. In order to get the tubes to go to the bottom, you have to screw the cap on and shake them kind of gently in order to get them to the bottom, in order to remove all of the air. I did 6 of these tubes. I added glucose powder, fructose powder, and mannitol. First, I'll just go over quickly the glucose, the Staph epidermidis, and the Staphylococcus cerevisiae. They have both fermented with gas production. You can tell by the yellow color that it was successful. The next 2 tubes that I did the same process, I added fructose. And here is my fructose added to by Staphylococcus epidermidis and Staphylococcus cerevisiae. And as you can see, they both have fermented with gas production as well. The last 3 tubes, I added mannitol powder, and we have a little bit different results here. Okay, on the Staphylococcus epidermidis, it just fermented a little bit. The Staphylococcus cerevisiae, I would say it's close to a non-fermentation. That would be this one right here, because it's very pink, close to red, which is the original color that I started with. Those little mini tubes inside, called the Durham tubes, it's what enables the detection of carbon dioxide and hydrogen gas. Basically, it traps the gas inside. And so what I have demonstrated is another technique for identifying unknown microorganisms by fermentation, by adding a dry chemical to my tubes, and also adding the pure organisms of the Staphylococcus cerevisiae and the Staphylococcus epidermidis. As you can see, I will bring them in close, turn them around so you can see the colors and the end product. Again, my name is Billy Dunn. Today is June 6, 2018. This is my lab exercise video for biochemical testing for microbial identification by carbohydrate fermentation. Thank you.